In this video, we're going to look at some of the genome informatics capabilities of the Pathway Tool software. The first thing we typically do in Pathway Tools is to select the default organism that we want to work with. So we're going to select the Prochlorococcus Mrna PGDB that we created in the previous pathologic video. We simply select it from the list of organisms in the Pathway Tools starting screen, and now it's our current organism, and we get various statistics on this PGDB. Let's now enter the Genome Browser from the chromosome menu, and we're looking here at the region in red of the full genome sequence shown here at the top. Genes in the same color are in the same operon, and with the Genome Browser we can zoom in or out to see more or less detail. We can also move left and right. You'll see the red box indicating our position change as we move along the genome. We can use this navigation area also to change our zoom level, and we can zoom down to the sequence level here, for example, or out to a much higher level of detail. We can also zoom all the way out so that we're seeing essentially the entire genome in one screen, where each of these little blips is one gene. The genes are not drawn to scale here. Let's actually go back to the starting screen and select the E. coli database so that we can show a few other capabilities of the Genome Browser. Notice that in the EcoCyc Genome Browser, we're seeing additional types of sites that we have data curated for, like promoters and terminators and pseudogenes and transcription factor binding sites will be shown in some cases. Now, one thing we can do with E. coli is we can turn on the Genome Browser tracks capability. What we've done is, using the command add external track, we have imported a GFF file containing predicted genomic islands or regions of foreign DNA in the E. coli genome. And those are indicated, those predicted genomic islands are indicated using these horizontal bars here, and as we zoom in or out we see that information at different levels of detail, and we can also move right and left in the genome as well. Let's now go back to Prochlorococcus, back to its Genome Browser, and let's turn off the tracks. And if we now click on a gene from the Genome Browser, we go to a gene page. Okay, this is the gene page for the ARG-H gene, which lets us retrieve the protein sequence or the nucleotide sequence of the gene, or any larger arbitrary region of nucleotide sequence within the genome. We can also go back to the Genome Browser. We have links to various other databases. Then we have a summary of each of the one or more functions of this gene. In this case, the gene product catalyzes two different related reactions. Now we have a number of queries available for proteins, for RNAs, and for genes. Let's do a substring query now. And we can enter the substring TRP here, and we'll find in this genome all the genes that have TRP in their name. Let's select them all. And what happens when we have multiple selections is we now see the first one, the TRPA gene, and the others are all on what's called the answer list, and we can look at the next answer to see these other genes. If we look in the Genome Browser, we see that in this organism the TRP genes are not in a single operon, which they are in some organisms. Now, to show a few other queries, we're going to once again go back to the EcoCyc database, and we're going to here query for DNA or mRNA sites, because the EcoCyc database has many different types of sites defined. For example, there are attenuators, promoters, transcription factor binding sites, many different types of sites, and we can not only search for sites by type, but filter them by their map position, for example, or type of evidence available for them. But let's simply search for all attenuators. Here's a list of all the attenuators. Let's just select the first one. And so this actually takes us to another type of information page, the transcription unit page, which shows a detailed picture of the transcription unit containing the attenuator that we just selected, showing that there's a couple different promoters at this transcription unit. There are several transcription factor binding sites known. There are several terminators known. And if we scroll down, we'll see a detailed description of each of the sites that control that regulate this operon. If we now click on this site, it will take us to the transcription factor page for that transcription factor, and scrolling down, we eventually get a list of all the gene ontology terms assigned to that protein, as well as all the transcription units that this transcription factor regulates in E. coli. So as you can see, the regulon of this transcription factor is quite extensive. We then get essentiality data for knockouts of this gene from several known essentiality data sets, and we get sequence feature information, that there are mutagenesis variants of this gene known. There are several PFAM sites known for this within this protein. I'm not going to show you an additional type of protein query. We can query proteins by their molecular weight and their isoelectric point. We can also query proteins according to their regulation. And so we could choose proteins that are either activated or inhibited or both by a particular compound, or those that are sensitive to a given cofactor. Let's choose those that are either inhibited or activated by L-lysine, and we could choose a different compound here if we want. And the result is these 6 proteins. Now finally, let me note that if you run Pathway Tools in its web mode, there are a number of other search types available. In web mode, we can perform blast searches against any genome in Pathway Tools. We can do sequence pattern searches against the genome, and we can also perform sequence alignments.